Very sorry now, so probably gonna give you actually 5 minute production.
Okay.
Oh, feeling like a home. I really like this. Work. And then we're gonna be talking about
So. Here, you can hold on to that. Here, I'll turn that off. You're going to turn it off for now?
Yeah.
But if you all
Okay, so good morning and welcome everyone. My name is Jose Macali-Piero. I'm an assistant professor of biomedical engineering here at Columbia University. And before I introduce our speakers, I have a few announcements to give. Today, we continue to offer this lecture in a hybrid format with both virtual and live audiences. Our speaker will present for approximately 40 to 45 minutes, and we will leave 15 minutes at the end for questions and answers. For our virtual audience throughout the presentation, please use the Q&A feature at the bottom of your screen, not the chat, to submit your questions, and we will try to answer as many questions as possible during the Q&A session. For our live audience, we will have the chance to ask questions during the Q&A. So today, it's my honor and pleasure to welcome Jean Fan, Assistant Professor of Biomedical Engineering in the Center for Computational Biology at Johns Hopkins University. Oh, yes, sir. Her research team, the Jeff Works Lab, is interested in understanding the molecular and spatial contextual factors shaping cellular identity and heterogeneity, particularly in the context of cancer and how this heterogeneity impacts tumor progression, therapeutic resistance, and ultimately clinical prognosis. She develops new open source computational software for analyzing single cell multiomics and imaging data that can be tailored and applied to diverse cancer types and biological systems. The impact of Dr. Fan's work has been recognized by several awards, including the Forbes 30 Under 30, the Nature Research Award for Inspiring Science, and the NSF Career Award. So please join me in giving a warm welcome to Professor Fan.
Well, thank you so much, Jose, for the very kind introduction. And yeah, I'm so excited to be here to tell you a little bit about my lab's recent work. Okay, hopefully that's not too echoey. Maybe. Or maybe actually, well, anyways, great. Yeah, so just before I dive into things, I wanted to show you some of the friendly faces behind the work that you're about to see. And as Jose mentioned, we are a bioinformatics research lab in the Center for Computational Biology, and we call ourselves the Jeff Works Lab. And when we look at a tissue, a mammalian tissue, like what we might see in a mouse brain, for example, hopefully, let's see, is there a way to turn off the lights? Thank you, Lisa. Yeah, hopefully, yeah, I do a lot of black backgrounds, so perhaps having the lights off will be a little easier for y'all to see. Hopefully I'll be exciting enough so you don't fall asleep. But anyways, when we look at a tissue like the mammalian brain, we're very interested in asking questions such as, what are the cell types and cell states present within this tissue? How are these cell types and cell states spatially organized? How does this spatial organization relate to function? And ultimately, how does this spatial organization potentially differ in the context of health versus disease? So luckily for us, there are spatially resolved transcriptomics technologies now provide us with an excellent opportunity to address these questions. And what these technologies allow us to do is really measure what genes are expressed in cells or small regions or small collections of cells in the tissue while preserving their spatial organization in the tissue. And these technologies, they span many different forms. So for more information, I encourage you to check out our Nature Communications review. But broadly speaking, my hope today is to teach you all a little bit about, what are these imaging-based molecular resolution spatial transcriptomics technologies, and how do they differ from capture and sequencing-based pixel resolution spatial transcriptomics technologies. And ultimately, now, once we have all these technologies and begin applying them to different tissues, how do we begin making comparative spatial analyses, potentially across samples, but also across technologies. Okay, so yeah, let's dive into the imaging type of spatial transcriptomic technologies. These technologies effectively, they go by many different names, but effectively what they all allow us to do is take pictures of molecules, take pictures of molecules, and through many rounds of imaging, we can decode which molecules are corresponding to gene A, which molecules are corresponding to gene B, and of course do that for actually hundreds to thousands to up to now tens of thousands of different RNA species within fixed cells in tissues and cultures. And of course, we can do this not only for one cell, but for the hundreds of millions of cells that we have. And given this subcellular information, we can begin asking a lot of different fun questions. For example, we might be interested in how different RNA species are co-localized to different subcellular compartments within cells. So we can actually combine these molecular resolution imaging techniques with things like immunostaining, where we can stain, for example, the endoplasmic reticulum, take a picture, and then computationally segment out where the endoplasmic reticulum is and see which genes overlap with that endoplasmic reticulum and begin quantifying which genes are statistically more enriched in that endoplasmic reticulum component, for example. But of course, being an imaging-based approach, now we can take all these genes that we've identified. We can take all these genes that we've identified as statistically enriched in the ER and plot them back onto our original ER staining image and begin appreciating how each of these dots is a gene species that we identified as enriched. And indeed, it overlaps very nicely with the very fine mesh network of the ER. So since this is published work, I won't really delve too much on it and will refer you to the publication if you're interested in learning more. Other fun things that we can do with this molecular resolution spatial transcriptomic technology is to leverage certain biological knowledge that we have about how RNAs are born and transported in order for their life cycle to produce proteins and allow the cells to do things, and use that information to predict putative transcriptional states of cells in the future. So again, these cells are fixed, they're dead. There's, in theory, no way for us to follow them as they move. However, we can use our knowledge of biology and how RNAs are born in the nucleus, and in order for them to effectuate functions, they have to be exported into the cytoplasm, made into proteins, and so forth. We can develop differential equation models to model that dynamic and effectively derive differential equations that we can solve that allow us to predict then in the future, at a future time point, what genes might be upregulated or downregulated in each cell, and use this to predict for a future transcriptional state of each cell. And in this manner, project both the observed and future transcriptional states into a lower two-dimensional embedding and visualize that using computational methods to be able to begin predicting, again, how these cells may behave in the future, even though we have this current one static snapshot. So again, this is published work, so I won't dwell too much on it, and will refer you to our various papers if you're interested in learning more. But a lot of people, when they have this type of information, we don't necessarily have to focus on the subcellular compartments. We can, for example, computationally segment out each cell and count how many of each gene species is in each cell. And that allows us to achieve a truly, single cell resolution spatial transcriptomic measurement, where now each point is a single cell in this coronal section of the mouse cortex. But of course, because we've measured all these genes, we can count how many of each gene is in each cell, and we can compare how these genes are expressed across these cells and visualize the high expressed cells in red and lowly expressed cells of this particular gene, SLC17A7 in blue. And do that, of course, for all the genes that we have measured in this targeted panel to begin appreciating all the different spatial patterns of gene expression within this tissue. And of course, we can also do more computational analyses, such as dimensionality reduction and clustering, where we can identify cells that are transcriptionally more similar to other cells based on our targeted panel of gene expression measurements, and in that manner, identify different cell types. But of course, given that these are spatially resolved measurements, we can project them back onto the tissue and again, begin appreciating how these different cell types are spatially organized in the tissue. Okay, so that is a very fast run through imaging-based spatial transcriptomic technologies. I hope I've been able to show you a little bit about how these technologies can provide us with single molecule resolution detected, but for targeted gene panels of tens to hundreds to thousands of genes, this information allows us to characterize how these genes may be organized within subcellular compartments. We can additionally leverage this information to begin modeling RNA velocity and future transcriptional states of cells. And finally, with computational segmentation, we can achieve single cell resolution spatial transcriptomic measurements, again, for these targeted sets of thousands to tens of thousands of genes. Okay, great. So that is a very fast run through of the imaging-based techniques, because today I really want us to focus on more the capture and sequencing-based approaches. So again, these techniques allow us to measure how genes are expressed within tissues, but the way they do it is a little bit different. So rather than taking pictures, effectively what these technologies allow us to do is tag RNAs in the tissue with different spatial barcodes. So I suppose this is probably a little hard for you all to see, but essentially this is a grid array, and there are these little spots that might be hard for you to see piled along this slide. And then we can take a tissue, lyse the contents, the mRNA contents from this tissue. The mRNAs will hybridize to these little oligo-DT probes that are in these little spots. And each of these probes have associated with it a spatial barcode that's going to be unique to each spot. So what this means is that when we create our libraries and subject the slides to the sequencing, we will be able to, for example, this is a spot here. All the spots have the same spatial barcode associated with it. So in the future, when during the course of your experiment, when you create your library and subject the results to sequencing, we can, for example, pick out the part that has the mRNA, align it to a genome and figure out which gene that corresponds to. But of course, now we also have the spatial barcode. So by reading out that spatial barcode, we can figure out where that RNA came from in terms of which spot. So at the end of the day, what we often end up with is this spatial XY coordinate of each spot. And with each spot, we have associated with it a set of gene expression measurements that we obtain from sequencing. Yeah. And this, as you might begin to appreciate, really gives us untargeted transcriptome-wide profiling by sequencing. Everything that's all the mRNAs that can be pulled down through poly-DT, we'll be able to capture and sequence. So now each point is a spot. And again, what I'm showing is the high expression of this particular gene in red and low expression in blue. And again, we can do this not only for one or two genes, but for actually all the genes that we can decode in the transcriptome. However, one thing to keep in mind that is, again, we can perform clustering analysis on this type of spot resolution data. In this case, if we again do our dimensionality reduction and graph-based clustering to identify spots that are similar to each other, we can identify these clusters of transcriptionally similar spots and then project them back onto their original tissue. And you can kind of see various patterns. However, it's important to keep in mind that it's important to keep in mind that these pixels and spots may comprise many cells, and therefore the gene expression measurements that you obtain from these spots are going to be become from all of these cells. And as these cells may represent many different cell types, the gene experiments that you obtain from each spot is also going to reflect the mixture of those cell types. So why does this matter? Yeah, why does this matter? I think on some level, perhaps you're all very, very bright, very smart, so you can figure out on a theoretical level, even mathematical level, why this matters. But I often like to take a more hands-on approach for exploring why this might be a challenge. So let's do a in silico experiment via a simulation. So in this case, just to prove this point that this might present a challenge, I'm going to use a simulation. And for those who are more hands-on learners, you're welcome to check out our lab blog for the code associated with this tutorial. But effectively, what I'm doing is I've simulated a single cell single cells where each column here is a single cell. And you can see that there are 3 cell types here. Each row is a gene, and red is high expression, blue is low expression. And you're all smart enough to tell me that there's definitely 3 cell types here. And indeed, we can cluster these cells using principal components analysis and see there's 3 clusters. Great. Okay, so now this is without any spatial information, but let's create some spatial information. I'm going to take this tissue and I will create some spots on this tissue, just like we might have for our real spatial transcriptomics data. And I'm going to start seeding these single cells in these spots at different proportions. In particular, I'm going to seed the cell type A as being in this gradient pattern, and the cell type B, I'm going to seed in this particular frequency pattern along the x-axis of the tissue. And I'm going to seed cell type C in this particular gradient pattern, such that if I visualize the cell type proportions, this is the ground truth. This is what I've created. And of course, I can also create the analog spot resolution gene expression measurement by aggregating the gene expression of the seeded cells in these simulated spots. Okay, so now, this is typically what we would get. And I might hand this over to a student and I tell them, oh, tell me how many cell types are in this data set, and they will do their clustering. And in this class, and in a class, perhaps they'll do a dimensionality reduction on this multicellular simulated data and obtain the following principal component dimensionality reduction. And if you guys are all smart enough that maybe if you look at this, you'll tell me that it looks like there's 2 clusters. So you'll pick, cluster with you'll identify your 2 clusters and visualize how they're distributed in your tissue, and you'll get something like this. And then you might come back to me, it's like, wait, Professor Fan, you told me that there were 3 cell types. So I will go back to you and say, okay, why don't you cluster using some higher K and force it to give you 3 cell types? And you will find yourself obtaining 3 clusters and visualizing them on the tissue. But essentially what I hope you'll appreciate is no matter how fine you cluster, you'll actually never recover the gradient patterns that we created in the first place. And likewise, the clusters that you obtain and the average gene expression of those clusters are actually never going to recapitulate the original gene expression profiles we seeded in the first place. So this is where a general approach called deconvolution comes in. And in particular, my lab developed a deconvolution approach called ST-deconvolve to recover cell type specific gene expression and proportions within these multicellular pixels in a manner that doesn't rely on external single cell references. For those who are more interested in the math, I encourage you to check out our publication and read more about the underlying model. But for today, I will try to explain the model a little bit more using analogy. So briefly, ST-deconvolve, our tool, builds on this generative modeling approach called latent Dirichlet allocation. So it's a model that's actually originally commonly used in natural language processing for the purpose of topic modeling. So topic modeling in natural language processing is what we might do if we have, for example, a large collection of documents. Say we have documents from the New York Times, and we want to, each document is a collection of words. So for example, some documents might have 5 copies of the word spatial transcriptomics and 5 copies of the word single cell. And other articles in the New York Times will have words such as baseball and basketball and player or something like that. But these words, as you might imagine, are distributions, are effectively comprising different topics. So perhaps our spatial transcriptomics and single cell words are associated with the science topic, and our basketball and baseball words are associated with the sports topic. So what we're interested in doing is associating different topics with different documents based on their collection of words. And these topics are latent and unobserved, so that's what we're trying to infer. So perhaps based on analogy, you can now begin appreciating how in spatial transcriptomics, we can think of each pixel as an analogy to the documents, where each pixel is now a collection rather than words of genes. And these genes are representing different cell types. So what we're really interested in is inferring the latent representation of different cell types in different spots and pixels. And we're going to do that by using the distributions of gene expression. So what we get at the end of the day is, again, given this multicellular pixel resolution gene expression data, ST-deconvolve using latent Dirichlet allocation is able to deconvolve back out this, the putative cell type proportions per pixel or per spot, which we can represent as these little pie charts. But then we also recover the original, recover the deconvolved putative cell type specific gene expression profile. So if we go back to our simulation, if we applied ST-deconvolve to this multicellular pixel resolution simulated data, indeed we can recover this gradient of cell type proportions that is consistent with our simulated ground truth. And we can appreciate that both visually, but it's not just I matched up the colors that is more similar. We can also, of course, quantify the correspondence between the transcriptionally best match cell types in the deconvolved ST-deconvolve results compared to the ground truth. And perhaps indeed we can see that deconvolution is able to achieve, recover the expected cell type proportions in a way that clustering analysis is not able to. Okay, but of course, this is just a fun simulation, and it's always good to return back to our original real data here, where again, we can apply ST-deconvolve. and now recover the finer cell type spatial organizational structures within, for example, the hippocampus and within the ventricles in a way that is not as readily observed through clustering analysis alone because of the mixtures of cell types present in these spots. And yeah, and for more, again, for those who are more hands-on learners, I encourage you to check out our blog post, which you can have the code to reproduce all these results and see for yourself. Okay, and of course, in our paper, we apply SD-Deconvolve to quite a number of diverse spatially resolved transcriptomics technologies that have these spots at various resolutions, ranging from the hundreds of, I guess, 50s of microns to hundreds of microns, and show that we're still able to recover cell type specific gene expression profiles and proportions. So I encourage you to check out the paper to learn more. And of course, we are interested in developing these technologies and computational analysis methods, ultimately to better understand, again, the spatial organization of different cell types in the context of diseases such as glioblastoma. So this is just a sneak peek into some ongoing collaborations with Mihailina Janaszewska's lab, where we've applied this pixel resolution spatial transcriptomic technology to her glioblastoma mouse models, and we can deconvolve back out the spots that correspond to the glioblastoma tumor in red, but now also begin appreciating the gradients of immune cells that seem to co-localize around the tumor and begin also infiltrating the tumor, again, at these sub-spot proportions. And yeah, and perhaps next time I'll have to tell you more about it. But for now, I hope I've been able to show you a little bit about how capture and sequencing based pixel resolution spatial transcriptomics technologies are able to provide us with a full transcriptome-wide profiling, but at these pixel resolutions, which may comprise many cells and many cell types. However, deconvolution analyses, such as with SD-Deconvolve, can recover back these cell type specific gene expression profiles and proportions within these multicellular pixels. Great. Okay, and yeah, and for the last part of my talk, I've shown you these two different technologies, and actually I've showed them for approximately the same tissue. They are from different mice, but a question arises in terms of, how can we begin perhaps making statistical comparisons regarding, say, the molecular and cell type composition at matched locations across tissue samples. You're smart enough that you look at this tissue slice profile by the single cell technology, and you notice it looks somewhat similar to the one that was profiled by this pixel resolution technology. You can see similar hippocampus structures, dentate gyrus, and other brain structures. And perhaps you'd be interested in asking a question, especially if these samples came from different conditions. What are the gene expression differences at this particular region of the hippocampus in this data set compared to this one? And I suppose we could do that manually, but if we have lots of data sets to go through, we probably don't want to do that. We want to come up with some approach to align these two tissue sections and then begin making these 100 micron resolution comparisons on the aligned tissue space. So what we did was develop an approach called ST-Align to achieve this type of spatial alignment between different spatial transcriptomics data sets. And in particular, we build on advances in computational anatomy and this particular algorithm called large deformation diffeomorphic metric mapping, which is quite a mouthful. You can call it LDDMM for short. But to walk you through how this method works, and again, I encourage you to check out our bioRxiv preprint, which was just posted last week, and also our corresponding blog post, which tries to explain the underlying mathematics in a perhaps more approachable manner. But effectively, what ST-Align does is take these single cell resolution spatial transcriptomics data sets, which are often represented as cell positions, XY positions, and we convert that into a smooth image through a process called rasterization, which effectively, mathematically speaking, involves convolving the XY coordinates with a Dirac function with a Gaussian kernel. But effectively, what you need to know for now is that we take these points in space and we turn it into a picture that's kind of smooth. You can think of it as we plot it and we blur it a little. But once we have these smooth image representations of the two spatial transcriptomics data sets, now you can kind of see how this becomes an image matching problem. Effectively, we're trying to solve for a function that when applied to the source image, allows it to morph and become more like the target image. So we can solve for this function using diffeomorphic metric mapping. And I wanted to highlight some equations that might be a little bit challenging to digest, but I promise you it's not too scary. Effectively, we're trying to, again, whenever we're solving for a function that is trying to minimize the difference between these two images as captured by this matching term. So this matching term is going to be effectively 0 if these two images are exactly the same. So, and that's what we want. So we're trying to solve for a function phi that's going to, when applied to the source image, and we subtract it from the target image and subjected to a certain weight matrix that I'll describe a little bit more later, is going to be as small as possible, while also subject to a certain regularization constraint represented here by this R term. So again, we're trying to match the images as best as possible, but we need to be subject to certain regularization penalties, which effectively just means that whatever transformation we create has to be diffeomorphic, which also means that it has to be smooth. And there are many reasons to want this property. And the main reason being that we think that for tissues to morph into each other, we shouldn't have to kind of pick up this one tissue section and flip it all the way around, or pick this chunk up and put it all the way over here. So we want to keep our transformation function realistic. So we use this regularization constraint to impose that. And yeah, and effectively we're able to solve for this function using gradient descent. So through many iterations, it's as if we're rolling down a hill, and after 1,000 iterations, we'll find the bottom of the hill. And at that point, we solve for the function. And given this function, we can now apply it back to our original sets of XY coordinate points and transform those points into this new spatial coordinate that is aligned with our target spatial transcriptomic data set. So I think it's always fun to visualize the point movements as it goes through this diffeomorphic alignment, just to appreciate that it not only has this affine component, but also this more nonlinear local distortion component in order to achieve the alignment. Okay, so, and of course, in this particular case, again, with our original single cell resolution spatial transcriptomic data set versus our multicellular target, you can see that we have the source is only a component of the target. But that's okay, because again, we have this weight term that's able to actually divide our image into a component that is the matching component versus a component that's a background. So in this case, we can focus on aligning only the matching component, which is learned automatically. And then we can also see which areas of the tissue are part of this matching component and focus again only on aligning this matching component. And once we've achieved the matching, we will be able to begin comparing apples to apples. So this is what we obtain after applying ST-Align. This is the matching component of our single cell resolution spatial transcriptomic data set that's been aligned to the spot resolution spatial transcriptomic data set. And now we can begin making comparisons at these matched spot resolution locations, for example. We can look at genes, for example, that are detected in both data sets. And you can begin appreciating how even though we're measuring, again, these are two different mice, two different tissue sections, they've actually been subjected to different preservation techniques, but still we can see similar patterns of gene expression. In this case, high expression in the cortex, high expression in the hippocampus. And we can see how that compares when we have a single cell resolution measurement versus when we have these multicellular pixel resolution measurements. And of course, we can do this not only visually, but also quantitatively and quantify at each pixel what is the corresponding kind of groups of cells that have to be aggregated to create the same pixel size, and how does the expression of these pseudo spots in the single cell data correlate in expression with the expression measurements we get in the spot resolution technologies. And of course, do that now for all the genes we have access to. And other things, again, we can not only look at correspondence of gene expression, but again, for the single cell technologies, we can do our dimensionality reduction and clustering analysis to identify cell types. So now with the pixel resolution technology, we can likewise do a deconvolution using SD-deconvolve. And now we have cell types achieved from single cell clustering and cell types achieved through deconvolution. And we can evaluate how cell types correspond to each other in this aligned tissue space. Again, we can focus on a particular cell type and ask, at this matched pseudo spot, what is the proportion of this particular cell type in that spot compared to what we see based on deconvolution, and begin again appreciating how well the underlying cell types in space correspond across these technologies. And we can do that both visually and also quantitatively based on cosine similarities and other quantifications. And of course, we can do this all with tissues like the brain, but my lab is particularly interested in applying these to aligning serial sections of tumors, where, again, we might, in this case, these are two breast cancer sections, and you'll notice that they're partially matched with each other. However, we're still able to achieve a spatial alignment. In this case, I've also visualized the weights in the optimization function that I was mentioning, and you can see that we're able to detect certain parts of this first image as components in red that we should align, and components in black or blue that we've detected as the background. So we can just focus on aligning the red parts. And then there are also little green parts that the model has learned to be artifacts that will not contribute to the alignment accuracy quantification. And this is particularly helpful when we want to align, for example, single cell spatial transcriptomics to other imaging modalities like histology. So in the background here is a H&E stain of the breast cancer tissue section, and we can take spatial transcriptomics data sets from that same breast cancer section and align it back to the original histology and begin doing fun things with digital pathology, for example. But again, we can show that the weights associated with this alignment In particular, since histology images will also stain for things like things in the outside of the cell that might not be profiled by spatial transcriptomics, such as things in the extracellular matrix. These are things that we would not be able to align, but we're able to accommodate through our use of this weight function, so that we don't focus on aligning those things. Yeah, great. So all in all, I hope I've been able to show you that SD-Align is able to enable alignment of spatial transcriptomics data sets using diffeomorphic metric mapping. It's fun to think about, now that we have this tool, what can we do with it? What can we use it to do? Some things that we're very interested in using SD-Align for is actually just comparing across spatial transcriptomics technologies. We're interested in, as we move forward and applying these different technologies, we want to understand which technology has the better detection sensitivity for this particular dense region that we're very interested in characterizing well, for example, or perhaps which of these technologies have is able to resolve the more complete cell types and cell states that we're interested in characterizing. So comparing across technologies is one important application we're looking to apply this to, but also integration across spatial transcriptomics technologies. You've seen how one set of technologies gives you single cell resolution, but for targeted genes, and another set of technologies gives you spot resolution but full transcriptome. Well, why can't we use spatial alignment as a way of leveraging the best of both worlds? We can take serial sections, apply each technology to each slice, and then align them back together and use space as a way of integrating this information. Other fun things we can do is, once we have an alignment, is to do spatial comparisons of gene expression and cell type composition, as I showed. But of course, this becomes a lot more fun when we have, or a lot more interesting when we have perturbations and other physiological axes to compare across, both in terms of health and disease, but also in terms of perhaps treatments with different drugs. And other things looking forward. My hope is that these types of alignment approaches will ultimately allow us to accomplish 3D reconstruction through serial registration, but then of course also integration with multi-omics by applying different types of technologies against the serial sections. And yeah, I'm sure there's other fun things I haven't thought about yet, but I encourage you to think about it. Yeah, so with that, if you're interested in trying out some of our tools, you're welcome to check out our lab website. We have lots of tutorials and blog posts to try to teach you how to use the tools that I've spoken about today. And with that, I just wanted to thank, in particular, my students, Kaylin Clifton, Manju Anat, Lila Ada, Brenda Miller, and Feiyan Huang, whose work has been highlighted here, as well as our collaborators and all our funding sources. And for those who would like to keep in touch, you're always welcome to reach out to me via email at jeanfan@jhu.edu. You're also welcome to, once in a blue moon, probably find me on Twitter. And I'm also trying now to get onto YouTube. So with that, thank you all for your attention and happy to take any questions.
Okay, so thank you so much, Professor Sam, for an absolutely fantastic presentation. So before we get into the Q&A, I want to call everyone's attention to our last hybrid seminar of this academic year, which will take place next week on Friday, April 28th, with Zoran Nenevich from the University of California, Irvine. Please follow us on social media to receive the latest updates, and you can use our social media information on the slide. And you can always access the registration information for our BME seminars on our website at bme.columbia.edu. Okay, so we have questions to get through online, and I'm sure that our live audience also has many questions. If we do not have time to address your question, you can see your speaker's contact information on this slide. It's there. Okay, great. So please feel free to reach out following the seminar. Okay, so let's dive right into it. So let's start with the audience questions. Please.
So for the STL line, you said that you were using the contour of the tissue to match with the target image?
Yes, effectively we're using cell density and from both the source and the target tissues to achieve that line. So it's very reliant on essentially meso-scale structures and tissue sections of different.
And then the last one was so where did you say it's target image?
So you're trying to align to this vision as permanent.
Yeah.
One we call the source, one we call the so the target image is also achieved through the faster image approach. So now we raster both data sets and images and we can provide.
Okay.
Exactly.
Okay, got it.
If you already have an image to begin with, which is the case with the histology,
Yeah.
then you would skip that optimization step and along to the image.
Okay, so yes, sure.
So STL line seems really cool, and you did mention that you want to like solve this like integration problem. So like in particular in these imaging technologies, right, as you said, you don't get the full transcriptome. So could you potentially first like map it to the Visium, the capture and seek technologies, and use that to infer the full transcriptome?
Very interesting. Okay. Yeah, just to reiterate the question, Shovik, the idea is, can you use spatial alignment as a way of imputation effectively? That and yes, yeah, but again, it's worth keeping in mind that we are not aligning unless we're generally not aligning single cells to single cells. So the imputation would be achieved at some type of spot resolution at which your tissue has that variation in gene expression, which I fully expect will be very different for different tissues.
Okay, let's take one question from online from the Q&A. So this is from CUHE. Regarding the simulation, how to determine the cluster number? Why is the cluster number 3, not 2 or 4?
Yeah, that's a great question. I think, yeah, what's the optimal way to determine a cluster number? There are a number of heuristics based on within cluster similarity, which you want to minimize, and across cluster similarity or between cluster similarity that you might want to maximize, that you can use as guides to pick the optimal number of clusters. In this case, in the simulation, if you had done that, you would probably end up with a cluster number of 2. But yeah, I'd encourage you actually to try it out for yourself. What happens if you pick a larger number of clusters, and the extent to which picking a larger number of clusters will allow you to recapitulate the gradient structure, for example. It's a great question. And I encourage you to explore for yourself.
So another one from the chat for STL line. Is the registration mostly dependent on the tissue structure of images? And in the imaging-based technologies, oftentimes the FOV is only a small field of view. It's only a small region of the brain tissue. Wondering if it is still possible to align such ST images. Yeah, I think the first part of the question is very similar to our previous question. But the second part of the question, again, especially when we're working with large tissues like the brain, a lot of times, we might only have a small region of the brain profile, and we might need to align that to something bigger. So that is exactly what we're hoping to achieve with this a combination of partial mapping through those weight terms, is that we can achieve this type of
small to large alignment. Where it gets a little bit tricky is if both tissue sections that we're trying to align have tears or holes. If one of them has tears or holes, you can model that as an artifact or something that doesn't match. If both of them have tears and holes, my recommendation would be to find a third tissue section that doesn't have tears and holes and align both for that. Yeah.
So we can go. So here in the room, if we have any, I'll switch back and forth between the Q&A and all you. Yes.
We have this question that I don't know much about, but question about tissue sections having holes. Have we, like, what about instances of medical imaging, like MRIs or PET scans, aligning it to an actual MRI scan of that tissue?
Yeah, it's a really excellent question, and something we're very interested in doing is actually aligning these 2D thin tissue sections into a 3D coordinate space. I didn't really get into it today, but I encourage you to check out the website where we do that. And the way to do that is actually by performing the digital morphism on the 3D and trying to find a way to work the 3D such that we can create a slice through the 3D that aligns well with the 2D. So we can do this with a high resolution 3D scan of something like the brain. The challenge when we think about other types of imaging modalities like MRI, for example, or ultrasound, is resolution. So, if we think about here, again, we're trying to align based on these tissue structures. So if we have something like an MRI where I would characterize as very low resolution, but it has many other benefits. For instance, doing invasive and doing time component to it. But the resolution is quite coarse. So because of that, we might not be able to align at an accuracy that is at a resolution, at an accuracy level that would be particularly interesting for some of the biological questions we're interested in asking. However, perhaps in the future, somebody will develop much higher resolution than our own.
So there's only question has to do with the glioblastoma. I wonder if you considered, like, as you talked about the gradients that were around the thing that you were able to see.
Yeah.
I wonder if you thought about those being potential areas of selectivity, and how would we make a test?
Yeah, so we are particularly interested in this case, understanding how the glioblastoma cells, these actually they come from different genetic effects and they have different mutations. So trying to understand how that mutational profile recruits a different tumor microenvironment. So definitely interested in that interplay at the border, but also the infiltration within the cells. Yeah, it's a great question.
On that note as well, have you looked at the deconvolving molecular subtypes within?
It's a great question. Thank you. So again, because we are doing these deconvolutions in an unsupervised manner, what we're really leveraging is the spatial, the variation of gene expression across spots. And also the transcriptional distinctness of different cell types. So if a cell type is really transcriptionally distinct and it's very variable across spots, there's some spots with 100%, some spots with 50%, some spots with 10%. That's much easier for us to deconvolve out versus if you have a very subtle transcriptional state change. That might be present only in 1 or 2 spots, but very rare. That's much harder for us to confidently deconvolve back out.
Okay. I think that was my way of asking him to see like a subtype just being superficially restricted.
Yeah, I think all we want to see a lot more validation.
No.
But I think
Fair.
Deconvolution can help prevent you from.
So,
I have a question. It's sort of on the same topic, I think, but so when you did your simulation and said you say there's 2 cell types, but there are 3 cell types, and then so in in many tissues, you say like these are the outer hair cells, the inner cells, the pillar cells, like it's different cell types are defined like anatomic. And in this case, if you these cell types are really defined based on their gene expression, I guess. So it seems a little like it could be, if you said I want 4 different cell types, you get 4. And if you said I want 7, you get 7. At what point do you say those are really the cell types?
Yeah, it's a really interesting philosophical question.
Okay.
Yeah, like at what point do we say that this is a functionally distinct cell type and not just a subtle change in cell state that perhaps could go back and in between states? Yeah, I think it's a really tough question, and I think a lot of folks in the field are still grappling not only with that question of the deconvolution context, but in the clustering context in general. We can always cluster finer and finer to get more subtly different cell states, and you could get 2 clusters of cells that only differ by 1 union. Is that really different? Or is that perhaps, given various artifacts and limitations in our measurements, something just, as a result of stochasticity by chance, this group didn't express its gene?
So you sort of had to decide this much difference, and then by our definition. So.
Yeah.
So I think we can do, let's see, one more from the chat. So one question we have, can you tell, this might have to do with the cell segmentation. Can you tell a brain tissue whether the transcript is from a cell body or a process? And I guess how well do you resolve spatially transcripts?
Very, very good question. Just sort of repeat the question of, can you tell in a brain tissue whether the genes come from a cell body or a cell process, such as a dendrite, an axon? So especially for neurons and other highly polarized cells, there are actually genes in the cell appendages. And in the context, when we make these measurements in tissues, the answer for now is no. And the reason is we take these thin tissue sections of maybe 15 microns in thickness. So your cell body is 10 microns or so, but the processes can extend in all directions and they come off. So there's no guarantee that our thin section has these processes contained within them. Even if they do, again, we need some way to trace the cell and its processes, which I suppose we could do with immunostaining of axon and dendritic markers like tau and F2, which we have done in the context of cultured neurons. And again, there, when it's cultured, everything is growing on an even thin section, so we can, we don't have a sectioning problem, and we can image. We can combine with the stain that allow us to see that the cell body is indeed connected to this process and be able to segment that out. But it's still, again, going to require us to segment, which is definitely still a challenge. If you think about how these axons and dendrites are very, very thin, and cross with each other, it's quite so yeah, so it's definitely a challenge, but I encourage you all to think about.
Okay, so we are exactly on time. That's all the time we have today. I'd like to thank our special guest, Professor Dan, once again. We look forward to seeing our in-person and virtual audience at our STV seminar of the academic year next week, and enjoy your week.